Hello everyone, my name is Anish Tripudi, and today I'll be showing a research project which revolves around the effects of ocean acidification on barnacle feeding behavior and predator avoidance. To begin with, here is the introduction. Because of increased carbon emissions from factories, cars, planes, etc., marine organisms' lives are being threatened. This is because the oceans are carbon sinks, which means that they absorb a lot of carbon dioxide. And because we are emitting a lot of carbon dioxide, the oceans are acidifying and warming up. When the ocean acidifies, the pH level decreases. This process is called ocean acidification. One of the many effects of ocean acidification is the decreased lifespan of marine organisms, especially those with shells. And one disastrous effect of ocean acidification is harming keystone species, especially those with shells. Keystone species are species that greatly affect and define an ecosystem. So even if one keystone species become endangered, the entire ecosystem can decline or be greatly debilitated. This is because organisms depend on keystone species for food, or they depend on them for keeping population of other species in check. The reason why we focus on barnacles is that they are important to the environment by filtering waste from the ocean, and they are also food for other animals. So what do we know about ocean acidification? We know that increased acidity causes lessened cell growth for marine organisms that use calcium carbonate for shells. It also degrades shells, which means that animals have to spend more and more energy to repair the shell, but they might not have enough energy to reproduce, thereby decreasing the population and lifespan of the organisms. For example, if there are fewer barnacles, their predators' populations, which are the sea stars and dog whelps, their population will decline because of lack of food. The predators of sea stars will also die because there is less food for them, and this creates a cascading effect all the way to the top of the food chain. This can also affect humans' food and economic systems because industries and humans depend on marine life for food and fishing industries. So we know about how ocean acidification affects shells, but we do not know how it affects feeding habits and the ability to avoid predators. This leads to our question: how will increased ocean acidification affect barnacles' feeding habits as well as their ability to detect and avoid predators? In our tanks, we have two species of barnacles: semi-balanus balanoids, aka acorn barnacles, which are shown here, and tetraclita rubescens, which are volcano barnacles that are shown here. Acorn barnacles are small and white and cluster up in groups, but on the other hand, volcano barnacles are big compared to acorn barnacles, and they also occasionally cluster up. The barnacles are locally obtained at Mavericks Beach on Half Moon Bay, but we have to keep on going back to replace the barnacles that occasionally die over time. However, we do keep track of new barnacles. Now for the tanks. To keep the pH at the desired level, we have to add in CO2 every day. We have 3 tanks in total. 2 of them are experimental tanks that have CO2 added in every day, and these are the 7.5 pH tank and the 7.8 pH tank. 7.8 pH is the expected pH level of the ocean by the end of the 21st century, while 7.5 pH is to represent an extreme to see what happens. For reference, the control tank is 8.1 pH. The lower the pH, the more acidic the treatment is. We do water changes for each treatment tanks in case they get dirty, and each tank has volcano and acorn barnacles. In order to emulate the ocean, we use something called a live rock that contains lots of helpful bacteria, and we also use marine salt that has many helpful materials that are good for the barnacles. So how do we collect data? To quantify the effects of ocean acidification on the feeding habits of barnacles, we first have to add in the barnacle's food, which is zooplankton. Zooplankton are small marine organisms. And we add 1 pipette of zooplankton for each species of barnacle in each tank. Then we count how many times the barnacles extend their cirri. A cirri, right here, are these feather-like structures that help catch the zooplankton, and we count how many times the cirri emerges per minute interval. So For 1 minute, we count how many times the barnacles extend their cirri for every 10 minutes and until the 30-minute mark. For predator avoidance, we first put a barnacle in a small container with the corresponding tank's water. To imitate a predator, in this case a sea star, we touch the barnacle with a sponge on a stick. We record how long it takes for the cirri to retract after touching it with a sponge. After removing the sponge, we also know how long it takes for the cirri to re-emerge. We do have some sources of error because we did not add CO2 to the tanks daily. We also were not able to do feeding data collections every 3 days, which can affect graphs, and did not collect enough data on predator avoidance for an in-depth analysis. But with the start of a new semester, we are adding CO2 every day, data collections every 3 days, and we're doing more predator avoidance data collections. One thing to note here is that the results and graphs are preliminary, meaning that they're expected to change as we gain more data. First, we used R and RStudio. R is a computer language geared for data science, and we could do many things in R, such as graphing data, making box plots, and doing calculations with said data, and so much more. RStudio is an IDE made mostly for R. RStudio is a great platform with a good graphical user interface where you can edit code and run it directly to see the output, and some examples of outputs are graphs and tables. The graphs shown here are box plots, and box plots color in the central 50% of data points. Each box plot represents a tank, and each data point represents the amount of cirri extensions per minute for one barnacle. The line in the box plot is the median of the box plot, while the red dot is the average or mean of the box plot. The graph at the top left is a comparison of cirri extension for each tank. The graph on the top right is a comparison of barnacle cirri extensions, which excludes volcano barnacle data, and the bottom graph is the same as the top right graph, except it excludes acorn barnacle data. The reason why we made two separate graphs that exclude acorn and volcano barnacles is that volcano barnacles are less active than acorn barnacles. Results. The control tank has a higher range with about 0 to 30 cirri extensions per minute interval. The 7.5 pH tank's barnacles has 0 to 25 cirri extensions per minute interval, and 7.8 pH tank barnacles have 0 to 23 cirri extensions per minute interval. The average and median of the control tank are higher than the rest, and the range and median of the 7.5 pH tank are higher than the 7.8 pH tank, even though this does not mean it is statistically significant. The top graph here shows a comparison of cirri extensions excluding volcano barnacles, and the bottom excludes acorn barnacle data. So these graphs show that volcano barnacles are less active than acorn barnacles, and the mean and range of cirri extensions per minute interval of the control tank are higher than the rest. And we see this in the past graph, and it shows a similar pattern to the past graph as well. For p-values, we use analysis of variance, or ANOVA test, to calculate a statistical difference. P-values are one of the many ways to view the significance of data, and if a p-value is lower than 0.05, that shows statistical significance. The p-value of the control tank and 7.8 pH tank is 0.003, meaning that there is a significant difference. The p-value between 7.5 pH tank and 7.8 pH tank is 0.0223, meaning a statistical significance. One thing to note here is that the barnacles in the 7.5 pH tank are much more active than the 7.8 pH tank, even though that the 7.5 treatment is more acidic. There is no statistical difference between the control tank and the 7.5 pH tank because the p-value is 0.2085. What does this mean? Using current data, 7.8 pH treatment harms the feeding activity of barnacles compared to the control tank, and the 7.8 pH treatment also harms the feeding activity of the barnacles compared to the 7.5 treatment. The second bullet point here is interesting because the 7.5 treatment is more acidic. So in theory, 7.5 treatment should harm the barnacles even more. The control tank has a higher range and mean, which shows that the barnacles in the control tank are more active, and there's no significant effect between the 7.5 pH treatment and the control tank. We have two main future goals. We want to first automate adding in CO2, or at least try to add in CO2 more frequently. We also want to do more data collections, especially predator avoidance, which we don't have enough data for graphs. Our goal is to have a month and a half of daily CO2 inputs and more data collections in order for a more in-depth analysis. And finally, we want to thank ASDRP and sponsors for giving us the opportunity to conduct research, and we also want to thank our advisor, Andrew Benson, for helping us. I also want to personally thank current and past researchers in our group. So here are the references, and here are the image references.
Wonderful job, Anish. It's awesome. All right, does anyone have any questions?
Yes, I have a couple. I have a couple of questions about the control tank. Where did you, okay, how was the control tank created? How was it constructed? Did you just take seawater, or did you mix something? How was the control tank fixed up so it was delineated as a control tank?
So for all the tanks, we get regular tap water, we add dechlorinator, and we also add marine salt. The difference between the control tank and the experimental tank is that we do not add in CO2 to the control tank, whereas we add in CO2 for the 7.8 pH tank and the 7.5 pH tank.
Okay. Did you measure the pH on the control tank?
Yes, we do. It's 8.1 pH. We have a pH meter, which we use for all the tanks.
So 8.1. It might be useful to indicate that on your slides, what the pH of the control tank is.
You put it here.
Okay.
Does anyone else have more questions?
Yes, I do have a question. Why ANOVA, why not chi-square test?
Can you repeat your question?
Yeah, why you use ANOVA test rather than chi-square?
We use ANOVA test because that was like the one provided for us, and it was like the fastest, in my opinion, because
What do you mean by fastest? It's not that like they use a different engine.
ANOVA test was really the only test I personally knew, so that's why we used it.
Well, since you knew only one, doesn't mean that that's the only statistical analysis you're going to do, because with ANOVA, you are comparing the means. However, you are comparing the difference between variable of control and experimental. So in this case, looking at chi-square and putting the hypothesis that there's a significant difference between control and experimental is the one that's suitable for your studies rather than ANOVA, because you're not looking at the difference of two means, because one of them is control. So that's why you got significance, because again, like you're comparing the mean. If you use chi-square test, it's what exactly you needed for your analysis. So it's just because you just know ANOVA. How come you know ANOVA but you don't know chi-square?
I mean, that was suggested to us by Mr.
Yeah,
Benson.
I can.
Oh, Andrew. Okay.
Yeah.
Okay.
We started using ANOVA for that. That was what we just kind of got through for our data analysis. I am personally more familiar with ANOVA, and we were planning to use chi-square. We're just not done with data analysis. This is just preliminary.
Okay, gotcha. Thank you.
No worries.
All right, does anyone else have more questions? All right.
Oh, actually, I have one more question. Why do you think the results in the 7.5 tank were closer to the control than the ones in the 7.8 tank?
We don't have like a concrete explanation, but what we believe is that maybe the barnacles in the 7.5 pH tank is just naturally more active, but we need evidence to actually support that, and we're still trying to figure out why that is the case. And yeah, since the results are preliminary, they're expected to change.
Okay, so you're going to do more experiments regarding the variation from control for the different pH levels.
Yeah, we're gonna do more experiments overall. Yeah.
Are you going to try to drive the pH level down even further?
I don't think so because we're trying to keep it as like constant as possible. That's why we add in CO2 every day to keep the CO2 level, the pH level as like stagnant, you know. So we want to have the 7.8 pH tank to have 7.8 pH all throughout the experiment, and the 7.5 pH tank to have 7.5 pH all throughout the experiment.
So might it not be useful to have a setup at 7.2?
It might be, but currently we want to try to use just 2 tanks, but we're probably gonna, like, you know, maybe add on.
That would provide an additional data point for the variation with pH.
Yeah, that's true.
Okay, I'm done.
All right, so does anyone else have more questions? If not, I'll stop sharing.
All right, I guess my group's up next. So yeah, I guess if Akshati, Shika, and Ashley could share.